morning, friends. I am Dr. S.P. Khairna from Manmad College. Today, I am going to discuss with you one topic from botany paper first of the F5BSc new credit system syllabus, and name of the topic is life cycle of Riccia. Riccia, it is a small plant and it belongs to group Bryophyta from plant kingdom. The bryophytic plants at present we remember them as amphibian plants or amphibians of the plant world. You know amphibian group that from zoology means animals which are living in aquatic as well as land habit. Similarly, in plant groups, this bryophytic group, it is very peculiar that they live at the area which is neither to aquatic nor terrestrial. That is in between terrestrial and aquatic habitat, that region where maximum moisture is present and land habit and moist conditions. So that we call it as a amphibian habitat. So bryophytic group, it is very typical, and as we know them as the amphibians of the plant world. The plant body of this Riccia or bryophytic plants mostly is very simple, and it is called thalloid plant body. Means these plants do not have true root, stem and leaves. So it is called thalloid plant body. Now let us study the classification of the Riccia. Riccia, it belongs to division Bryophyta. Because why it is classified into bryophytes? Because it has thalloid plant body, that is simple plant body, no true root stem. Then vascular tissue, you might be knowing, that is xylem and phloem, water and fluid conducting tissue, we call it as a vascular tissue. And this vascular tissue is also totally absent in this group Bryophyta. So we have classified Riccia in bryophyte because it is having these characteristics. Second, Riccia, it belongs to class Hepataceae. The reason for classifying Riccia into class Hepataceae is as Riccia has thallus, leaf-like, green chloroplast it is having, and that chloroplast is typical, it is without pyrenoid, and the fruiting body, or what we say the sporophyte in Riccia, it is in the form of single capsule, and that capsule does not have columbella, that is sterile cells which are present in higher bryophytes. They are not present in Riccia, so we classify them into the class Hepataceae. Then Riccia belongs to order Marchantiales. The diagnostic characters of this order Marchantiales is that there are scales are present on the thallus of the Marchantial group. The scales, these are the violet colored bodies present on the ventral side of the margin of the thallus, and it is considered that these scales, they are responsible for withholding the water, moisture, which is very essential for the reproduction stage in the Marchantial group thallus. So that scales are present. Then rhizoids in this group Marchantiales, there are two types of rhizoids present, that is tuberculate and smooth wall rhizoids. Then another diagnostic character of Marchantial is air pores and air chambers are present. These are the diagnostic features on the basis of which this group is recognized. Then Riccia, it belongs to family Ricciaceae. Here in this family Ricciaceae members, sex organs, they are present at dorsal groove, that is on the upper side of the thallus, and sporophyte with capsule only. Means sporophyte of most of the bryophytes, especially higher bryophytes, they have foot, seta and capsule. But Riccia, it belongs to primitive bryophyte or lower bryophyte, so it is having a very simple sporophyte, that is in the form of capsule only, and foot and seta, they are absent, and genus Riccia. Second point in the life cycle of Riccia is about the habit and habitat. Habit and habitat. Here the Riccia genus is represented by about 200 species from the different parts of the world. At present, this Riccia genus, it is distributed all over the world. However, it is observed especially in the southern hemisphere of the globe. From India, about 31 species of the Riccia are recorded, which are found mostly in the moist places like banks of the streams, wet rocks, walls, tree trunks, etc. Similarly, in Maharashtra, Riccia species they are observed at Saptashringi Hills, Salner Fort, Lonavala, Mahabaleshwar hill stations, etc. Next point, external features of the Riccia thallus. How externally this Riccia thallus looks in the nature. When we go to the nature, how we can identify the Riccia. Here the vegetative plant body of the Riccia is small gametophyte, which is a dominant phase in the life cycle. That thallus, it is flesh green, flat and dichotomously branched. Very typical character of Riccia. It is the thallus is dorsiventral and dichotomously branched. Here scales and rhizoids which are present on the ventral side of the thallus. Scales are multicellular, violet in color and present on the either margin of that dorsiventral thallus. The rhizoids which are present on the ventral side of the thallus, there are two types, smooth walled rhizoids and tuberculated walled rhizoids. The smooth walled rhizoids, they are long, unicellular, while tuberculated rhizoids are having pig-like outgrowths inside the wall. The rhizoids perform the function of absorption and anchorage, means they absorb the water and minerals from the moist soil and provide support. Anchorage means support to that thallus to survive. So this is the structure of vegetative structure of the Riccia thallus. See here, it is having dichotomously branched structures, and also in the figure you are observing the tuberculated walled rhizoids, smooth walled rhizoids, and the violet scales which are present on the ventral side of the thallus. Now we will go to study the internal structure, anatomical structure of the Riccia thallus. When the dorsoventral thallus of the Riccia, when we take the transverse section of that thallus, then you will going to observe that anatomically the Riccia thallus, it is divided into two regions, that is on the dorsal side photosynthetic region and on the ventral side or lower side the parenchymatous storage region. Now what is photosynthetic region or assimilatory region? Assimilatory region, it is chlorenchymatous. It means chlorophyll pigments are present, so automatically this region is concerned with synthesis of food, that is photosynthesis. It is made up of vertical rows of chlorophyllous cells and they are separated by air chambers. There are many rows of chlorophyllous cells and separated each row from other by the air chambers. Each that vertical row contains about 6 to 8 green cells, and the topmost cell of that row, it is colorless or hyaline. These colorless upper cells forms the upper epidermis. The air chambers, they are the means of gaseous exchange, that is O2 CO2 exchange. We know the chlorophyll pigment or chlorophyll cells during the daytime when they synthesize the food by taking carbon dioxide from the air and they produce oxygen. That is O2 CO2 exchange takes place visually through these air chambers. The main function of this region automatically we know it is photosynthesis, that is the synthesis of food. So this is figure, it explains the anatomical structure of the Riccia thallus. The upper epidermis, single layered, each cell is separate. Then the photosynthetic region having the chlorophyllous cells arranged in the rows. Then towards the lower side, you will find the storage region. Storage region, it consists of multicellular parenchymatous cells, and each cell is with plenty of residual food material in the form of starch. That is, the cell's photosynthetic region synthesize the food material, and that residual food material, it is stored in the storage region. The lowermost cell of this region form the single layered lower epidermis, and on this lower epidermis the tuberculate rhizoids and smooth wall rhizoids are present. Now the next point, reproduction in Riccia. Riccia, it reproduces vegetatively and sexually. That is, it have both methods of reproduction, that is vegetative method of reproduction and sexual method of reproduction. During the favorable periods or favorable season, that means during the rainy season, for example, when all the conditions are favoring for the growth of that plant, during that favorable period, this plant reproduces by vegetative method of reproduction. But at the end of growing season, when the rainy season ends, at that time the moisture is not available, humidity is not available, water is not available, so that plant starts to produce sex organs, and by that way that plant reproduces at the end of the season by sexual method of reproduction. The vegetative method of reproduction, in this method any vegetative part of the plant, it have ability to develop new Riccia thallus, because every plant cell have a capability of totipotency. Totipotency means total potential. Single cell or group of cells or part of the organ or the plant body is able to develop the entire plant. That fragment of the organ or single cell or the tissue, when it is cut off naturally or due to some accident, then that cut part of that tissue or that cell is able to develop the new entire plant, what we call the totipotency. So the vegetative method, it is based on the totipotency capability of the plant cells. First method from the vegetative method, it includes fragmentation. Fragmentation means when due to death and decay of the older parts of the thallus, which leads to separate the dichotomous lobes, and each lobe as it gets separated due to death and decay, and now each lobe is able to grow as a separate new thallus. So this is what we call fragmentation. Second method, it is by tubers. In some species like Riccia discolor, the tubers are developed at the margin during the unfavorable conditions of the environment, and these tubers, they grow, swell off due to plenty of residual food material stored in them, and after sufficient growth these tubers, they get separated from the mother thallus, and when favorable conditions prevails, these tubers, they will grow into the new independent Riccia thallus. Third method of vegetative reproduction, it is called by adventitious branches. This method is observed only especially in the aquatic species, for example Riccia fluitans. In this plant, some adventitious branches are formed on the ventral surface of the thallus, and these adventitious branches, they separate and grow as new thallus. So that is reproduction by development of adventitious branches. Now the fourth method of vegetative propagation is by development of persistent apices. Persistent apices, these are growing down in the soil from the apical region of the thallus, and they become thick and store plenty of residual food material. When favorable conditions prevail, Then these persistent apices, they grow into the new independent Riccia thallus. So friends, we have studied these four different types of methods of vegetative reproduction which are commonly observed in the Riccia. Now we'll go to the sexual reproduction. Sexual reproduction in Riccia, it is called as a oogamous type. There are three methods of sexual reproduction: isogamous, anisogamous, and oogamous. In isogamous type of sexual reproduction, both the gametes, that is male and female gamete, they are of equal shape, size, and both are motile, so we call them as a isogamy. In still some other group, we will find anisogamous type of the sexual reproduction. Here one gamete is small size, one gamete is large size, but both gametes are motile, and if union of such unequal gametes takes place, we call it as a anisogamous type of sexual reproduction. And this third method, that is oogamy, here one gamete is very small size, it is motile, no doubt, and the second gamete, it is large size and it is non-motile. So when this one small size motile and one large size non-motile gamete, when they unite, then that method of sexual reproduction is called as a oogamous type of sexual reproduction. And this method of sexual reproduction in Riccia, it generally takes place in the unfavorable conditions, that is at the end of the growing season, when the environment conditions gets dried up, humidity, moisture content get lowered. So at that situation, plants start to develop the new sex organs. The sex organ, they are well developed in Riccia. Male sex organ, it is called antheridium, and female sex organ, it is called archegonium. Plant may be monoecious. Monoecious means when antheridia and archegonia, when they are produced on the same thallus, then we call that species as a monoecious thallus. While in some other species you find that the male antheridia, they are produced on the separate thallus, and archegonia, they are produced on the separate thallus, so they are called as a dioecious species. As vegetative thallus bears sex organs and the physiological, all the processes, they are going on in the thallus, so this phase we call it as a gametophytic phase or gametophyte. Now let us study the structure of male sex organ, that is antheridium. Antheridium, which represents the male sex organ, the mature antheridium of Riccia, it is oval to pear shaped. Look into the figure, that antheridium, it looks like the oval or pear shaped. It has short stalk at the base, you will find that short stalk at the base, and club shaped antheridial body. The antheridial body is covered with outer sterile layer of jacket cells and central mass of androgonial cells. Antheridium, mature antheridium, it is covered with antheridial chamber that you are able to see in the figure. The androgonial cells in the center, that is shown by green color, they mature into the male gametes, that is called anthrozoids. Each anthrozoid is having unicellular structure and two large motile flagella. The anthrozoids, they are liberated free from antheridium when the conditions are favorable, that is moisture content. When there is a plenty of moisture content, then from that antheridium, the jacket and other structures break, and the anthrozoids, they are liberated from the antheridial body. At the time of liberation, jacket cell disintegrates and to ooze out the androcyte, that is male gamete, and this gamete get, or they swim in the semi-fluid layer. In this water film, they swim for a while, and finally they reach to the female gametophyte in the vicinity on the same thallus, that is archegonium, for fertilization. Each anthrozoid has typical two motile flagella. So this is the structure of antheridium. Now structure of archegonium. The archegonium, it is a flask shaped structure. See the figure. Typical flask shaped structure. It also develops on the in the thallus tissue. It has three major parts, that is stalk, venter and neck. Stalk, it is at the base. It provides attachment of the archegonia to the thallus tissue. Venter represent the basal swollen part and it encloses the female gamete, that is egg, that red color egg cell. Neck is a long, single layered, elongated tube and narrow canal. So the neck is shown long, elongated. And that neck consists of 6 to 9 tiers of elongated cells arranged in 6 vertical rows. Each mature archegonium is enclosed in archegonial chamber. Like antheridial chamber, here archegonia is also enclosed by archegonial chamber. At maturity, the neck cells and venter cells, it disintegrates to form a siphon-like canal, central tube, central canal. And the mucilage in the neck canal cells, which is formed due to disintegration of the above cells, that mucilage absorbs water, swells and separates the upper cover cell top. Through this open neck, the male gametes enter into the venter for the fertilization. So this is about the structure of archegonia. Now, after development of male and female gamete or liberation of the male gamete, that is anthrozoid, and it is reaching through the neck canal to the venter of the archegonium. Now this anthrozoid, flagellate anthrozoid, swim and enter through the mucilage neck canal, and finally it reaches to the venter of the archegonium, and in that venter it reaches towards the egg cell or the female gamete. Now here, when they come together, that what we call fertilization. Fertilization, it takes place in the presence of water. So very typical for the fertilization in the Riccia, presence of water is must. If there are dry conditions, if there is no moisture content in the atmosphere, then no fertilization will take place. So presence of water, moisture, it is very essential for the reproduction or fertilization in Riccia. Water acts as a medium for swimming of the male gamete, that is anthrozoid. Anthrozoid swim in water and reach archegonial neck, enters through the neck canal and reaches to the egg and fuses with the egg cell and develops the diploid structure, zygote, or what we call the 2n. Diploid, why it is diploid? Because n number of chromosomes, they are coming from the male gamete and n number of chromosomes coming from the female gamete, that is anthrozoid and egg cell. So when these two unite, fuse together, so it will form 2n, that is diploid zygote. And this diploid zygote cell, now it divides and redivides and finally it develops the sporophyte. Here from this diploid zygote, new generation in the life of the plant starts. That is what we call the sporophytic generation. The zygote, it will divide, redivide, it will develop the sporophyte. Sporophyte again, finally it will lead to produce the spores, what we call, by the reduction division, and again the gametophytic generation will start. The mature structure of sporophyte of Riccia, as already I mentioned initially, the Riccia sporophyte is very simple. It is represented by only globular capsule without foot and seta. So foot and seta, these are the two parts of the sporophyte and they are absent in Riccia. So only in Riccia you will find only the capsule. Firstly, the zygote, it will produce the wall around it. It will increase in volume. The ventral cell divides periclinally and anticlinally to form the calyptra. Then large number of spore mother cells develop in the capsule, which are diploid, no doubt. Then each spore mother cell finally at the last stage they will divide by meiosis or reduction division. And so the spore mother cells will divide meiotically and it will produce 4 haploid spores. So here from zygote to the spore mother cells, the sporophytic generation very limited, and the spore mother cell immediately undergoes meiosis and it produces 4 haploid spores. And these spores, they are present in the capsule. That red color structures, they are called what we call the meiospores or spores, and that capsule is observed in the figure. Now these spores in this way which are produced in the capsule, they represent the first stage of gametophytic generation. Now structure of spore and spore germination. The mature spores in the capsule, though they are mature, but they are not immediately liberated from the capsule, but they are retained in the capsule even a year of the period. That is, somewhat 1 year maybe the spores retained inside the capsule, though they are able to germinate, though they are able to develop the new plant, but they are not dispersed. Spores liberated from the capsule only by decomposition of the capsule wall and the surrounding calyptra. So the capsule wall and calyptra wall, once they get disintegrated or they get decomposed, then and then only these mature spores get chance to disperse from the capsule, and they get dispersed or separated, and then if the surrounding conditions are favorable, that is moisture and other conditions, then these dispersed spores, they will germinate and to produce the new gametophyte, that is new thallus of the Riccia. The fully mature spore is uninucleate, pyramidal shaped with 3 wall layers. The mature spore, for example, it is observed in the figure, it is having the triple layers: exosporium, mesosporium and endosporium. So spore germinate by rupture of outer 2 layers, that is exosporium and mesosporium. The endosporium, it protrudes, it comes out in the form of germinal tube, just like the pollen germinate to form the pollen tube in angiosperm. Here also in Riccia, the spore germinate by protrudence of endosporium. The tube germ tube divides to form 6 to 8 celled structure, what we call the protonema or primary thallus. The protonema, it further redivides and to develop the new thallus of the Riccia. Now we are coming towards the last part of the Riccia life cycle, that is life cycle pattern in Riccia. First, imagine the thallus, Riccia thallus, vegetative thallus, which is dichotomously branched, grow in the rosette form, many Riccia thalli growing together and forming the rosette. That Riccia thallus, it grows, produces food, develops scales, rhizoids, etc. It matures. Then this thallus, when it matures at the end of the growing season, now it will start development of sex organs, that is Antheridia and Archegonia. Antheridia, they represent male gametangium, and Archegonia, they represent the female gametangium. Antheridium body at maturity, it encloses androcytes or anthrozoids, what we call male gametes. While Archegonium also, it will produce the female gamete called as a egg cell. Here, from mature thallus to the development of Antheridia archegonia and towards development of the male and female gamete, that is anthrozoid and egg cell, this is the gametophytic phase. This phase It is called dominant phase in the life cycle of Riccia because the physiological phenomena like photosynthesis and other, all these phenomena they are taking place in this phase, in the mature thallus. So it is the dominant phase. Development of gametes, that is antheridia, anthrozoids and egg cell. Now in the next stage, this anthrozoid and egg cell they will fuse to form the diploid structure, what we call zygote. 2n diploid, because one n number of chromosomes coming from male gamete and n number of chromosomes coming from female gamete, they fuse together, so it will form the diploid structure called as a zygote. So the formation of zygote, it represent the first stage of the next generation, that is porophytic generation, because antheridia, archegonia, then the gametes, all they are haploid, but these two unite to form diploid. So this diploid stage, that is zygote stage, it is very typical in the life, and it represents the first step of the porophytic generation. Now this zygote, it divides firstly to develop 2 cells zygote or 2 cell structure, then again they will divide 4 cells, then 8 cells, 16 cells, like that. So such a multicellular structure which is developed from zygote, all cells no doubt they are diploid, and in this way it forms the small young sporophyte. That young sporophyte again finally it get converted into the mature sporophyte, and we know the mature sporophyte of Riccia, it is simple and it is represented only by capsule. Inside that capsule, which is all cells of the capsule, no doubt they are diploid. Inside that capsule, the spore mother cells which are present, they are also diploid. Now at the last stage of the sporophyte development, that spore mother cells from the capsule, they undergo meiosis or reduction division. Means each spore mother cell, it will divide meiotically and it will produce 4 haploid spores. So these haploid spores which are produced from spore mother cells, now they will represent the first stage of the gametophytic generation. Here sporophytic generations starts from the formation of diploid zygote and it ends by formation of the spore mother cells. Spore mother cells divide meiotically and it will produce haploid spores. So such haploid spores, when they are dispersed, when they are liberated from the capsule, then when they are coming in the vicinity, in the moist conditions of the soil or other area, then the spores they will start germination. When they will start germination, first initially it will form the small multicellular structure what we call the protonema. So protonema is nothing but the young thallus. That protonema cells, on the its ventral side it will develop small rhizoids, that is smooth wall, tuberculated wall rhizoids. Then also they will develop the what we say, the scales, and this division will, multiplication will go on, and finally it will develop the mature thallus of Riccia, which is dichotomously branched. Such a many thalli they are going together finally, and they will form the rosette structure on the even tree trunks or on the walls, etc. So this thallus in this way it will be developed from the spores. So when by studying this entire life cycle of Riccia, you will be able to see that that life cycle of Riccia passes through typically 2 distinct phases: gametophytic phase and sporophytic phase. For example, in the figure you will find the gametophytic phase which starts from the spore germination and it ends by formation of the gametes. And the major physiological phenomena which happens in this phase, for example photosynthesis, respiration, etc., all are taking place in the vegetative thallus, and vegetative thallus is haploid because it develops from the haploid spores. So this phase or the gametophytic phase, we find it is the dominant phase in the life cycle of the Riccia. While the second phase, what we call the diploid or sporophytic phase, the sporophytic phase starts from zygote, that is union of male and female gamete forming the zygote. it is diploid, and it ends by formation of the spore mother cells. So here this development of zygote into this sporophyte, that is capsule and spore mother cell, all they are coming developing on the cost of gametophyte. That is within the gametophyte these development phases are taking place. So sporophytic phase is depending upon the gametophyte. Sporophytic phase is not independent. It is not separately growing, but its developmental stages they are going on developing within the gametophyte, within the thallus. So sporophytic phase is depending upon the gametophyte phase, and gametophytic phase is independent. So this what the phenomena when we observe the entire life cycle, one phase follows the other phase again follows the first phase. So what we call the alternation of generation, that is one generation or one phase of the life cycle passes through the another phase, and one phase follows the other phase. So it is called, or they are alternating with each other to complete the life cycles. So that is called alternation of generation. So one generation alternates with the other generation to complete the entire life cycle. So for example, if any one generation is absent, can you imagine the life cycle will continue complete? No. For example, if sporophytic generation is absent, no new spores will be produced, no new thallus will be developed. So there will be no possibility of gametophytic generation. Or just imagine if the gametophytic generation is absent, then what will happen? If the gametophytic generation is absent, no sex organs are produced, no gametes are produced, so no formation of zygote will be possible. So both the generations, gametophytic as well as sporophytic generation, both are depending upon each other and they alternate with each other to complete the life cycle of the Riccia. So that phenomena what we call alternation of generation. So this is the very simple representation in the life cycle of the Riccia. So I express thanks for this. So I thanks to all for listening this lecture. Thank you.